All right, hello everybody. I am doing the 2025 NHT exam. I'm just going to go through the MCQ to begin with. So it's about 40 questions. Correct answer scores one. Yep, so in pencil. Okay, yep, all good. I'm not drawn to scale. Cool. Um, yeah, look, sometimes they might change the instructions, you never know. So I think it's always good to keep in mind, you know, the instructions, right? Cool. Yeast is used to help make bread. Okay, so they've changed the amount of yeast. All right. So they've used teaspoons, which is very interesting. Okay, so they haven't actually used any grams or anything. And then their bread height. So obviously Amar's probably be using less bread, but as long as he's been keeping it consistent, that's all good. So the amount of yeast is the dependent variable. It's not, it's the independent variable. Okay, so that's the one that we're changing. All right, the dependent variable would be the bread height. The height of the bread is the independent variable. No, it's not. It's not. So the independent variable is the other one. The amount of yeast is the controlled variable. It's not. The height of the bread is the dependent variable because it changes due to the different amounts of yeast present. That's probably the easiest question of all time. Okay, so obviously the height of the bread is changing, so that's the dependent variable, and obviously because of the variable that's changing, which is the amount of yeast, it is changing. When zero point 0.5 teaspoons of yeast was used, the broad height was consistently lower than when 1 teaspoon of yeast was used. This shows that, um, okay, so if we can see here, zero point 0.5, it's always lower. The data is quantitative, as a smaller amount of yeast would use a small amount of oxygen. Well, that is not true because yeast doesn't require oxygen, so that's not true. The trend is reproducible. Okay, they say and Amal's data showed both show this correlation. Yeah, so reproducibility basically refers to the ability of different people being able to actually perform this experiment. And the fact that two different people have been able to actually perform this experiment with similar data, right, shows that the trend is reproducible. The data is qualitative as each trial indicates that a smaller, so it's not qualitative, it's quantitative, right? We're actually getting real numbers. The data is not repeatable. Um, so just as I said before, just because Amal's data is showing a lower bread height doesn't mean that the data is not reproducible because he would have just used a lower amount of bread or like bread mixture, whatever. Cool. Okay, so below it shows the structure of hepatitis C. Just so this shows the structure of a hepatitis C virus. The virus is a single-stranded RNA virus, so it's single-stranded. So which one of the following diagrams could represent a repeated structure within molecular S within molecule S? So obviously they're referring to the RNA, I believe. The RNA virus, as we know, it's single-stranded. So definitely no bases, right, connecting with each other. Amino acid and tRNA, no. So we're talking about the repeated structure. So we only want to deal with the actual structure of the RNA itself. This is talking about tRNA, which is involved in, the production of proteins and whatnot. So that's fine. So I think we're looking at C because amino acid would not be applicable in this situation. RNA is the building blocks, is the instructions for the production of amino acid. So that's definitely wrong. But yeah, so RNA, they're single-stranded, okay, but each nucleotide tends to be made up of a base, a sugar, and a phosphate. Pre-mRNA molecules contain exons and introns. Which one of the following correctly summarizes the similarities and differences between exons and neutrons? And introns. So introns only do not leave the nucleus. That is correct. Okay, they do not leave the nucleus. They are subject to post-transcriptional modification. Both introns and exons, they contain nucleotides, but they are not translated. So that's wrong. They do contain nucleotides, but introns aren't translated. Introns only, I don't think they only contain nucleotides. So that's also wrong. Because exons also contain nucleotides. So if this is wrong, that's wrong. A would be correct. Okay, so exons are translated. Both introns and exons contain nucleotides, and introns only, they do not leave the nucleotides. I mean, they do not leave the nucleus. Which follower, which correct, okay. Which flowchart correctly orders some of the steps in protein synthesis? Okay, cool. That one looks pretty good. So transcription occurs in the nucleus, then the proteins are modified in the Golgi apparatus. I think the only thing in this situation is that they've skipped a couple of steps, but that's okay. The primary structure of the... Oh, wait, that's wrong. So we've actually gone from 1, 3 to 2. So 3 comes first. So we actually determine the primary structure of the proteins first during translation, right? When we're actually producing the individual amino acids. So that's definitely wrong. mRNA leaves the nucleus. mRNA leaves the nucleus, then processing occurs. No. Okay. First, we do the processing, then we leave the nucleus. Nucleotides are joined together. Okay. Then a poly-A tail is added to the pre-mRNA during RNA processing. Exocytosis of the proteins occur. So exocytosis basically means that the protein is leaving the cell. So how can it be modified afterwards? So it definitely would be D then. So yeah, pre-mRNA synthesized in the nucleus, then translation occurs, then two amino acid chains are joined to form the quaternary structure. Yep. So we've got a tertiary structure, tertiary structure. They can form bonds to form a quaternary structure. And then obviously the proteins are packed into circulatory vesicles for transport, for export, sorry. Three separate solutions contained the same substrate in either enzyme A, B, or no enzyme. All other variables were controlled. The graph below shows the amount of product produced in each solution over a 20-minute period. So enzymes A and B act as catalysts in reaction. Okay, that's not terribly wrong. Enzyme B is denatured between 15 to 20 minutes. Not necessarily. So we can see here that its function is still that like it's still producing product, right? So that's enzyme B, and it's still producing product. The reaction only occurs in the presence of the enzyme. That's also not true because we're able to still have the reaction go ahead and produce some sort of product. So that's also wrong. The active site of enzyme A is less complementary to the substrate than that of enzyme B. Okay. That's not necessarily true either, right? Enzyme A could just be more, oh, sorry, the active site of enzyme A would definitely be more complementary, right? The reason as to why is because the substrate is able to bind, the substrate shape must be more complementary to enzyme A, which is why it's functioning better. Okay. So it's would be the other way around. So it'd definitely be A. Cool. We are on to, wait, yep, 7. Cool. Um, So we've got a bit of gel electrophoresis. So they can be caused by genetic mutations. I did not know that. So identify if they carry the specific genetic mutation for T1DM. Okay, let's do this. Children with that T1DM. So they start off, okay. They from mothers from the well, and any other combinations of fragments result in children being diagnosed with T1DM. Okay, each of the lanes represent a different child being tested for the genetic mutation. Lane M is the DNA base pair ladder. The results are shown below. Okay, referring to the gel run, all the children carrying the specific genetic mutation for T1DM are shown in lanes. Okay, let's go through this. So, all we need to see here, okay, so if we go back to identify the character, children without T1DM have fragments of all of the same length that run furthest from the well. Any other combinations of fragments? Okay, so we just want, okay, the, mm, okay, so we want the kids that have their own sort of fragments all the same length. So that we want the ones that have all the same length. So this is the same length. This is the same length. Oh, so there's one child here that has just only one fragment here, another fragment there. That's it. That's all they have. So because 3 and 6, okay, have fragments that are of the same length and run furthest from the well, right? I'd be inclined to say that 3 and 6 actually don't have type 1 diabetes mellitus. All the other um, children actually have, right, this mutation. So this fragment. And that fragment would mean that it'd be 1, 2, 4, and 5, which is C. So, oh wait, no, maybe not. So two, okay. Hmm. Furthest from the well. I see, I see. Sorry. I've gotten it the other way around. Okay, cool. So, hmm. Sorry guys. So I think what I've done is I've done it from the other way around, but that's okay. So what I meant to say was that sorry, that it's going from here to here. I thought for some reason, because the numbers were there, my bad. I should have seen from the BP. Anyway, so sorry, it's running from this to this, right? Because the heaviest fragments stay at the start, right? Because they have the biggest, they have the most difficulty in traveling through the agarose shell. So the ones that that run the furthest from the well are this one, this one, this one, and this one, right? But what you don't want, you just want this fragment to be isolated and at the very end. So the ones that don't have type 1 diabetes mellitus would actually be number 4 and number 1 because they only have this fragment. They don't actually have the other fragment that would indicate that they have type 1 diabetes mellitus, right? Even though these people, like number 5 and number 2, they also have this fragment here, they also have this fragment there, and that does not run the furthest from the well, okay? So given that they only have this fragment that runs furthest from the well, which is 4 and 1, all the others would actually have diabetes, which is actually not C, it's actually D. Apologies. To prepare the DNA for profiling type 1 diabetes mellitus using gel electrophoresis, it is most likely that the scientists would have used. So that's a pretty easy question. So polymerase chain reaction, not necessary. Oh, yep. So they would have definitely, yep. So they would have used polymerase chain reaction for sure. Okay, and preparing the DNA for gel electrophoresis. So most likely we would have probably used endonucleases to cut up the DNA and then polymerase chain reaction to um actually maximize the amount of DNA that we have. Because when we actually use DNA, we don't have that much of it. So what we want to do, right, is we want to maximize the amount of DNA that we can actually have. Okay. Insulin, we wouldn't really use insulin in this situation. Plasmids and CRISPR-Cas9, that's a whole different topic. And I don't know why we would use ligase. So endonuclease to actually cut up the DNA, and of course polymerase chain reaction to actually, yeah, increase the amount that we actually have. So acetyl-CoA binds with the enzyme citrate synthase in the Krebs cycle. A competitive enzyme inhibitor that has the same shape as acetyl-CoA will most likely decrease the rate. So it'd be B. So it would not bind to an allosteric site. That'd be a non-competitive. It would have an effect. So it would most likely be B. The in cellular respiration, the net ATP yield from a single glucose molecule is affected by various factors, which from the following influences the yield produced during the electron transport chain. The rate of glycolysis in the cytosol, that would not affect it. Okay, the concentration. So the glycolysis in the cytosol, we're looking at immediate factors, right? We're looking at the electron transport chain. This is referring to the very first steps in the breakdown of glucose. The concentration of pyruvate, so that's again looking at one of the middle steps. So like the Krebs cycle, for example. So that's not it. But the main thing we use oxygen for is the electron transport chain, all right? So we use the oxygen. So the oxygen basically you know, we use it as it's basically a waste collector. That's the way I like to think about it. It collects, your H pluses that are left over after the sort of transport. Okay. So we definitely need oxygen. Okay. And if we don't have oxygen, it would immediately affect the electron transport chain. That is why during anaerobic respiration, when we don't have oxygen, the rate of glycolysis goes up, right? Which doesn't produce as much ATP. What could explain the differences in ATP yield between aerobic and anaerobic respiration? So the electron transport chain is not using anaerobic respiration, leading to lower ATP production. That sounds pretty good. Okay, so as I was explaining before, the electron transport chain requires oxygen. Okay, so in anaerobic, so an meaning non, and aerobic meaning oxygen, we wouldn't have oxygen, right? So A is pretty good. Anaerobic respiration produces more NADH compared to aerobic respiration, which decreases ATP yield. Um, no, not necessarily. So NADH are our carrier molecules. So aerobic respiration produces more NADH. The Krebs cycle is more efficient. No, it's not. The Krebs cycle doesn't even occur. Um, Cool aerobic respiration doesn't produces more ATP molecules. So A is pretty good. The decrease in the rate of glycolysis within a healthy muscle cell is most likely caused by, again, this seems to be very interesting, but in the decrease in the rate of glycolysis within a healthy muscle cell, hmm, yeah, so a decrease in the rate. It seems a bit odd, but I think, yeah, so the reason why glycolysis would occur within a healthy muscle cell is if it's being deprived of oxygen. Okay. So, um, so it's if it's being deprived of oxygen. And so I think the concentration of oxygen, okay, would mean that, yeah, I think so. So it should be, yeah, see, so that would mean that, yeah, where's my tab? Cool. Yeah, so the concentration of oxygen would result in the decrease in the rate of glycolysis. Cool. The two reactions shown below are the part of the biochemical pathway occurring in the same stage of photosynthesis in a plant cell. Where in the cell would the two reactions be happening? So, where is it that the like water is broken down into its hydrogen ions and oxygen gas? Well, that's mostly happening. So obviously the plants are producing oxygen gas, and then we're turning the hydrogen ions into NADPH. That's actually happening in the guarana, right? So in the actual parts, um, so in your little pancakes. So in the that's called the grana, or the granum, and then the plural of granum is grana. Cool. The limiting factor for this stage of photosynthesis would obviously be light intensity. Okay. So this is definitely the first stage, okay, of photosynthesis. This is the light dependent stage. It's happening across, and if I were to, so in a mitochondrial membrane, that's like, I think that's to do with more with like respiration. But if you guys were to remember across, so where the mostly it's happening, um, if I remember, like sort of across the membrane. Okay, and the water is basically being broken down into hydrogen ions and oxygen gas. The main reason water is broken down is so that we can get these hydrogen ions. Okay, and these hydrogen ions get turned into NADPH, which become our carrier molecules, so that we're able to facilitate the Calvin cycle. Okay, so it's very important that we have light. Okay, one of the roles of water in photosynthesis is to, I would say in this situation, act as a carbon source. Not really, okay? H2O, it's not really a carbon source. Convert directly into glucose, it does not. Provide electrons for reactions, yes. So if you think about it, when water is being broken down, we've got O2, yes. We've got H plus, yes. But what we also have um, are four electrons that are produced every time water is broken down. Now that might require a bit of chemistry knowledge. I don't know. I did take the chemistry route with that just then. But yeah, whenever water is broken down, we're getting the oxygen gas, but that's just used by us humans. But the electrons and the H pluses, they are actually used as facilitators in the next couple of reactions. Cool. Okay, cool. So light dependent reactions use ATP to produce glucose. So light dependent does not produce glucose. The light independent convert ADP into carbon dioxide and water. That is more respiration. Okay, the light independent reactions produce ATP from carbon dioxide and water, which is used in the light dependent reactions. No. Cool. So the light dependent reaction happens first. The light dependent reactions produce ATP, good, which is used in the light independent reactions to help fix carbon into organic molecules, good. All right, what are we fixing carbon into? Glucose. Okay, but it doesn't necessarily have to be glucose. It can be other stuff as well. Okay, what are the uses for glucose? All right, the plants can use them. We can turn them into cellulose. Why? Because then it can make up the plant wall. Okay. So it's a good idea to realize why, all right, and for us to eat as well, right? So the plants can themselves can use it. The glucose can be turned into cellulose, or that glucose goes into the fruits that we eat, right? Apples, stuff like that. Before an allergic response occurs, histamines form a chemical barrier. They don't really form a chemical barrier. Plasma B cells produce IgE antibodies, which then bind to receptors in the plasma membrane of mast cells. So before an allergic response occurs, that is true. So it's producing IgE antibodies, and they basically prime these mast cells, right? So now these antibodies are on these mast cells like little sticks. And so say for example, if I like for example, I'm very alert, like I get very much hay fever. Okay. So which is surprisingly also the next question. So I get hay fever quite often. So basically what happens is in response to that, when I breathe in the pollen, the plasma B cells will produce Ig antibodies and they will just bind to the mast cells. Now, if I breathe in pollen again, then the pollen will just basically bind to these antibodies and which will result in the release of histamine, which then causes all of our itchy um which causes all of our itchy symptoms, you know. Cool. Histamines are not found in peanuts. They're produced by our body. They do not pass through a physical barrier. No, that is not true. Uh, Blood vessels dilate in response. Yes, so it does. And that's why, basically, like if anyone's ever swollen, right? So say for example, these are our blood vessels normally, and then basically it basically enlarges, and what it causes is fluid to actually go from here. So it actually causes fluid from the blood vessels to go into our tissues, which is why sometimes in very extreme situations we get like, swollen areas. Complement proteins directly kill pathogens? No, they indirectly kill pathogens. So they actually assist in the killing of the pathogens. Okay, so it's very important that they've used this language. So it's indirectly. So it's true, true, false. Macrophages phagocyze the pollen. That is true. So 18 should be B. Lymph nodes are important organs of the lymphatic system. During an immune response, the lymph nodes produce plasma cells that release antigens. They don't really release antigens, they release antibodies. Attracted bacteria that are engulfed by, I don't know if they're attracting bacteria. Okay, they're not really doing anything. They're more the site of something. Oh, for example, the site of clonal selection expansion of B cells. Perfect. Okay, they are not releasing allergens. Okay, we are the ones that are breathing in the allergens, right? But basically, what happens is, as you guys know, the macrophages will come. Okay, you can think of the lymph nodes as like the head control center. They'll come to the control center, which is the lymph nodes, and they'll be like, hey man, there's like a whole situation going down. We're gonna need some B cells. And then those B cells convert to like plasma B cells, stuff like that. Q. Developing, which one of the following? Okay. Which one of the following graphs would be consistent with the antibody concentration in a person who is developing active immunity to the same antigen they're exposed to on day 0 and then again on day 40? So They're exposed to it on day zero, then they have a bit of immunity, and then they're exposed to it on day 40, and then boom, their antibody concentration goes way up. Okay. So that one looks not too bad, but this one does not look so good because at day zero, the antibody concentration is already up, and then it somehow goes down. That does not look good at all. So this one probably looks the best. So C probably looks the best. The reason as to why I've gone for C as opposed to A, even though they look pretty much the same, is because it's pretty unreasonable for the antibodies to actually remain. So the only differences between A and C is that little downward trend. And in C, that's a bit more realistic. So basically, what happens is we get exposed to it on day zero, and it takes about 10 days for our immune system to start building up antibodies. They'll start building up antibodies. And obviously, because these antibodies, they're just floating around, they're not doing much, they'll start to decrease, but they still retain some memory. So that when we get exposed to the antigen again, boom, it shoots right up, okay, and then it's able to give us it's able to increase its antibody concentration, okay, which is how vaccines work. Obviously, this is probably really unlikely because it's not like on day 10, suddenly it's really impossible for our immune system to just go from 0 to 100 in one day. The disease, Middle East respiratory syndrome, is caused by a viral pathogen. It's caused at bats with the initial host. Person-to-person transmission can occur. The steps in transmission are shown below. So it goes from bat, okay, bat to then camels. And then we eat milk and meat. And then it goes from humans to humans. No specific antiviral medication. The most effective strategy, okay, would be heat treating milk and fully cooking camel meat. Um, yeah, possibly. The removal of bats from the forests. So the most effective strategy from non-human to human populations. So we want to go from non-human. So from camels, we want to make sure that camels and humans aren't getting the disease transmitted. No specific antiviral medications exist. And generally speaking, for viral illnesses like this, antiviral medications they don't actually do much, right? We want something to prevent the transmission. This will just help treat the transmission. Allowing people to visit zoos that keep both bats and camels, obviously not, right? Because we want to minimize contact between the two. So C and D both look pretty decent. Okay. However, it seems pretty barbaric to just remove bats from forests. Okay, and it's not very effective. Okay. However, what we can have full control over is treating milk and fully cooking the camel meat before we actually consume it. Effective vaccine, 1963, 1980 can be the reasons be given with that previous infection could, as there was no carrier or the humans where the recent outbreaks of measles could be attributed to increased human contact with animals known to carry the measles virus. There is no animal carrier of the human measles virus, so that is wrong. Improved immunization programs in countries with high cases of measles. They're talking about outbreaks, and this is called talking about improved immunization programs, so definitely not. Increased mask wearing and use of disinfectant when in hospital settings. Interesting. Reduced measles vaccine uptake during and after the COVID-19 pandemic. Most likely the true answer. Okay. So during the COVID-19 pandemic, we will less people were taking the measles vaccine because everyone was obviously in their own homes. So yeah. Monoclonal antibody, obviously, it looks a little bit like this. What does it look like? Looks something like that. Mm, something like that. So there's four chains, so it definitely doesn't have one polypeptide chain. Would contain amino acid monomers. That is true. Has codons containing three nucleotides. So monoclonal antibodies are made up of polypeptide chains, which are made up of amino acid monomers. This is DNA. Okay, so this would be more like RNA. Okay. Has a tertiary structure as its highest hierarchical level? No. Okay, so this is how a monoclonal antibody looks like. Okay, and then it has its little active site there. And as you can see, there's two long chains and two short chains. So there's actually four chains, which mean it would be this. Also, another way that you can find out, like this is really interestingly worded. Because we can only pick one option. These two things are saying the exact same thing. Having one polypeptide chain and having only a tertiary structure are the same thing, and they both can't be true because we can only pick one option. So instantly, we can remove A and D. Now, obviously, we're not dealing with codons. I mean, we're not dealing with DNA, so it's not B. It's obviously C. Cool. When used in the treatment of an autoimmune disease, monoclonal antibodies. Okay. Attached to viral infected cells. So we're treating autoimmune disease where our human body, um, our body um attacks our own tissues, okay? Suppresses an immune response. So yeah, so what's basically happening, okay, in an autoimmune disease is um the immune system is mistakenly suspecting our self tissue to be foreign. And so what we want to do is suppress that immune response. Obviously, that leads to, increased chances of infection because our immune system isn't working as well. So it's a bit of a catch-22. We don't want cytotoxic T cells to attack self cells, so that's definitely wrong. Increase the activity of antigen-presenting B cells. We also don't want our immune system to keep going up, up, up. And a new immunotherapy strategy is being developed. Chimeric antigen receptor T cells. Okay, so you remove blood from patient to get T cells. Mm, Okay, so basically what we're doing is we're basically inserting the gene, okay, of CAR, and CAR basically breaks down the DNA, breaks down the sort of cancer cells. So a gene inserted to CAR T cells must code for the production of antibodies. So I don't think they're coding for the production of antibodies that act against cancer cells, but what they're doing in this situation, if I got it completely right, is that they're actually producing CAR T cells themselves. So what we're doing by inserting the gene is basically inserting um the what do we call them? The instruction sheet, the instruction manual to produce the CAR T cells themselves, so not the antibodies. CAR T cells replace all other T cells. We're not replacing other T cells, so that's not true. Ideally, that would be great. Okay, but for the successful treatment, we don't need it to replace all of the T cells. Receptors on the CAR T cells must bind to a specific antigen on the cancer cells. So that's perfect. So what we want is we can see that these CAR T cells are basically binding, okay, to specific antigens. So you can see these little horns of the cancer cells. They're binding to them and therefore destroying them. CAR T cells act as phagocytes and engulf the cancer cells. I don't think that is what they are doing in this situation. They're just binding to the cancer cells and killing them. I don't know if they're phagocytosing them. So that is wrong. Which two antibiotics show no change in percentage of resistant bacteria for the two time periods? Okay. So they show no change. Well, it would definitely be this one and this one. So wouldn't it not be 26A? So as you guys can see, there's very little change. So this one is 00, and this one's still the same. Based on the information provided, what is the following like? Which of the following is most likely a consequence of antibiotic resistance after continued antibiotic treatment? Use of only tetracycline will reduce. Which of the following is likely consequence? Hmm. Which of the following is most likely a consequence of antibiotic resistance? The use of only tetracycline will reduce the. So, in the stem, it says that we need to use a combination, right, of antibiotics. So you don't want to only use tetracycline. Use of only metronidazole and amoxicillin will not change the likelihood of patients developing ulcers and stomach cancers. So only metronidazole, okay, and amoxicillin. Maybe. I don't know. So amoxicillin will not change the likelihood. Yeah, it's likely. I don't know. This is okay. We'll come back to it. Use of only metronidazole and clarithromycin may increase the occurrence of ulcers and stomach cancers in the human population. So if we have a look at metronidazole and clarithromycin, yeah. So use of only levofloxacin and clever will reduce. So we don't want to use only that. So the only thing, so what it's basically saying is that if we use antibiotics by themselves, okay, the H. pylori infections will continue to occur. And the only one that actually supports that is C. Okay, so it's saying that, yeah, if we only use metronidazole and clarithromycin, it will increase the occurrence of stomach ulcers and stomach cancers happening. Ocean temperatures are rising, threatening coral reefs. The program heat tolerant corals for our reef recovery. The program involves identifying heat tolerant parent corals, collecting spawn. So eggs and sperm, mixing them, fertilization occurring from juices some heat-tolerant offspring, exposing and letting those are not heat-tolerant, testing survivors. Okay, so what they're doing is they're doing fittest survival of the fittest. If the program successfully aids reef recovery, the long-term effect on the coral gene pool would be obviously if we're basically creating something in the lab, that every single coral will be heat tolerant, all right? And therefore, the genetic diversity would actually go down. So increased genetic diversity due to the inclusion of a wider range of parent corals, not necessarily, because we're actually eliminating those that are not heat tolerant. Unchanged with the focus on maintaining existing, not really. Okay, because we're only looking at yeah, we're not looking at genetic variation here. Increased genetic diversity again, no. So I think would be D. So increased occurrence of alleles that provide heat tolerance. Yes, because we eliminating every single one that's not heat tolerant. It is important to test the coral offspring under ocean conditions in order to optimize breeding methods by identifying heat-sensitive coral strains early in the process, determine whether laboratory testing can be replaced by ocean testing, ensure that offspring can survive natural environment conditions, accelerate the adaptation. So we're not really trying to accelerate the adaptation of corals because we've already indicated that they can actually, be fine in the lab. What we're trying to do is try to ensure that these plants can actually survive, okay, in their own natural environment. We're also not trying to optimize breeding methods. That's not going to alter whatever things that we have from the coral strains. It's not going to alter the breeding methods. We've already identified the heat sensitive coral strains. We're just making sure that they can actually survive. So what does absolute dating? Oh, okay. So this is just pure recall. Okay. So obviously, as you guys can may be able to remember, all right, is even when we die, we leave a bit of a footprint because we're carbon-based organisms. Okay. And so absolute dating relies on the decay of radioactive elements. Okay. Obviously, relative dating is when we compare two fossils amongst each other, but the decay of radioactive elements is we're looking at the ratio um, of, yeah, the amount of, yeah, the ratio, something like that. So like 13 carbon to something, 12 carbon or something like that. But obviously, in the fossils, it would be different. Paleontologists were uncovered fossils. Okay. So, um, hmm. So these were all uncovered within the same layer. And this leaf was pretty much here to here. So this leaf was present from here to there. Okay. This was also this leaf thing looking was present from here to here. So I'm trying to look at the commonality between all of them. And then this was also common from here to here. So it'd be in the Paleogene period. So we just have to look at what's overlapping, right? So I'll just do that again really quickly. So if we look at the commonalities, so all of these four fossils were uncovered within the same layer. So we just need to find, one period in which all three of these are actually in the same period. And as you can see in one, two, three, as if they were all present here. One, two, three, four. Hmm. Okay. Oh wow. Birds rely on having unless it makes an indigo birds usually. Hmm. If some female indigo birds lay their eggs in the nest of a different bird species, the consequences could be that over time, it is likely that the offspring would. Yeah, so what they basically said is that they said that the indigo bats, they're basically it's just talking about like the environment that you grow in. You basically acclimatize to that environment. So even though the indigo birds are indigo birds, they still learn the five finch mating song because that was the nest that they were grown in, that they grew up in. So if we have female indigo birds that lay their eggs in the nest of a different bird species, okay, they would learn that new bird species songs. So A and D are wrong, leading to reproductive isolation and sympatric speciation. Learn that new species songs that only mate with five-finch birds, leading to allopatric speciation. Yeah. So, oh wait, sorry. No, it would not lead to allopatric speciation. It would lead to sympatric speciation. Okay. So I got these two mixed up, but there doesn't seem to be any geographical separation between these two species. So there would be reproductive isolation, okay, because they would only be reproducing with the five-finch birds, for example, and sympatric speciation because there doesn't seem to be any geographical barrier between the two species. Which one of the following correctly states shows the methodology? Um, so this would likely be field work. So we're not really doing a case study, blood sampling for hormone level analysis, not really. We're mostly observing behavior, right? So what are the indigo birds doing? Monitoring a reproductive success, not really. So we're not really counting how successful they are in reproducing and correlation study. Um, so that is a study that you could do, okay? However, it's not something that they've made very apparent. What they have made very apparent is, so in this specific study, what they've made apparent is they're just talking very qualitatively, not quantitatively, about how they sort of go about um their life. Cool. Um, okay, cool. Um, the upper phalange the upper phalange in the dogs may be considered a sample, may be considered an example of. Mm. So foot bones in the toes are called phalanges. Um, so the upper phalange of the dog, so that corresponds to the big toe in humans. So what could that be referred to as? Hmm. Um, yeah, so I would like to say that this would definitely be homologous structure. Okay, but don't know if it's molecular. And I'm not sure if they're vestigial as well. So they're basically anatomically the same. So they've got the same anatomic origins, but obviously they've got different functions. So they're homologous. But I'm not too sure of the vestigial. So vestigial means that they don't have as much function anymore, which I'm not sure if I believe in. So I'm not too sure about this. Maybe someone can help us out. I'm inclined to say C because it's not structural homology. It's not molecular homology, and they're not selectively breeding. So I'd like to say C, but maybe somebody can have a look at the report. Let me know. Cool. Hmm. So what happened to our the foramen, referring to the unclassified skull of the hominoid? It is reasonable to conclude that the hominoid, right, was was probably so let's see. Yeah. So chimpanzee human divergence date. An unclassified was occurred from 7.2. So we've got something from 8 to 5, and then we've got something from 7.2 to 6.8. Okay, so they diverged somewhere between here. Australopithecus afarensis is 3.7 to 3 million years old. Hmm. Modern human and an unclassified skull, and then we've also got the chimpanzee. So this skull is definitely older than the Australopithecus, right? Because this is dated from 7.2 to 6.8. So not bipedal, as the skull is dated as at 7.2 to 6.8. Um, not necessarily. So we're not really referring to anything there. That's not really good justification. It is bipedal, probably not, just because um this could have been before, you know. So Australopithecus was bipedal, okay, but yeah, so bipedal, so it's probably not bipedal. Not bipedal as a position of the foramen magnum does not align with that either of the Homo sapiens or the afarensis skull. I'd probably be, yeah, I'd probably be inclined to say C. Uh, or maybe it is B. Hmm. Does not align. I do agree. However, this foramen magnum is actually in between that. So that actually could be. So actually, that's actually probably not true. Too sure. Bipedal as a foramen magnum is positioned towards the front of the skull. Oh, yep. Sorry. More similar to that of the, yeah. So I'd actually say that since it is actually closer, so since that foramen magnum is actually closer, okay. Like we know that this is bipedal. I know these are bipedal. And given that this is in between these two, it's probably going to be B. It's not towards the back of the skull. It's not similar to this. So it's more similar to these two. Sorry, misread the question. Cool. Hmm. Species X. Okay. Referring to the information is reasonable to conclude. Hmm. So we want something that is so so we've got Homo erectus. And they got similar, different, different, similar. Okay, that's 900,000 years old. And then we've got different, different. Okay, so yeah, so let's go through the options. Homo sapiens and species X are the most closely related, as the fossils were found recently. So Homo sapiens and species X are not the most closely related because they both have different characteristics. Should be classified as the same species as they both lived in Indonesia? No, because they still have different characteristics. Species X could be a putative species of Homo erectus, as they share similarities of both species. Um, no, um, there's not enough, like there's probably not enough evidence for it to just be a hybrid species. And there's no sort of evidence of interbreeding or direct evolutionary relationships from just like two anatomical features. So species X may have evolved from Homo erectus. I think may is a really important word here. Okay. Because it's saying it might be, right? Because the teeth are similar. That's a bit tough though. The fossils were dated around 700,000 years old. Generally speaking, the best way to date fossils is to basically find out the rocks, the dating of the rocks, and then use that relative. So date the rocks in which the fossil is located, and then use that to relatively decide where the fossils, like what time the fossils are from. And so the EDPR gene found on chromosome 7 helps contraction of the hand. Gene has many alleles. Some of the alleles have an origin. Okay. So the Neanderthals evolved in Europe before moving to Africa, as evidenced by the presence of this EPR1 allele. Um, no, there's no indication of Africa here, and they would have gone to Africa first, then Europe. And it's a very sort of strong statement. So this evidence of that doesn't support them going from Europe to Africa. There's literally no relation. This EPDR1 allele is more likely to have arisen due to a mutation in Homo sapiens. Um, Not necessarily. It's not very likely that we have a whole allele that's due to a mutation. Okay. There has to be some indication. This is more indicative that the Neanderthals fossil had this allele and so did we. So I would say that there is some sort of evidence that there was a bit of interbreeding going on with Homo sapiens. Are different species, therefore it is likely that they would have an identical DNA sequence. If there are different species, then it wouldn't make it more likely for it to have an identical DNA sequence. DNA sequencing of the human produce short tandem repeats, short-term variants. Okay. Aboriginal Australians diverged from South Asians. Aboriginal Australians diverged from South Asians. Hmm. So this is South Asian. So they would have diverged here somewhat, right? So that was 54,000 years ago, with no recent common ancestor with South Asians at 5,000 years ago. So what? So it's, yeah, I'd say this is 5,000. With no recent common ancestor, so they don't have any common ancestor. That is pretty good, actually. I like that. So yeah, so this is South Asians. This is Aboriginal Australians, and their most recent common ancestor is here. Increased divergence. So South Asians increased divergence around 27,000 years ago, and a new species of Homo sapiens may have evolved. 27,000 years ago, there's no evidence here for a new species developing. That is not true. They are more genetically diverse as they diverged around 50,000 years ago. So 50,000 years ago would have been here or even here. So um, they're not, okay, I get what they're saying. Sorry. Yeah, so they're not more genetically diverse just because they diverged around then. There's actually no link to that. Okay. They're more closely related to South Asians. No, they're not because Papua New Guinea's they have more of a recent common ancestor, so that comes first as opposed to that one. I think D is my best answer. When comparing the two studies, it was reasonable to conclude the results of the 2016 study do not represent the true value of the number of STR short-term memory as the true value was already determined in the 2002 study. Um, uh, no, I don't think so because we actually used improved DNA sequencing techniques. So it made the 2016 study would be more likely to represent the true value of the number of SDRs. In 2016, the data is more accurate than the 2000 as improved DNA sequencing techniques were used, probably. The results of the 2000 are invalid as the 2002 recorded number of STRs and 2016 study constructed a phylogenetic tree. Mm, it's not, that doesn't mean that it's valid, it's invalid. Um, they're not precise as different scientists, so that has nothing to do with precision. It says C is probably the best answer. Um, yeah, that was okay. That was not too bad of a yeah, it's not too bad of a of MCQs. Um, yeah. Yeah, so not too bad. Cool. A lot of like opposite statements, but it should be chill.